Cancer is a disease caused by mutations. A cell duplicates its DNA in preparation for cellular division called mitosis. The molecules responsible for this duplication of DNA are extremely accurate, but every so often a mistake is made. This mistake is called a mutation. But if mutations occur at such a low rate, how is it that a cell can accumulate enough of them to become a cancer? To answer that, we need to take a closer look at the cell cycle.
The ultimate goal of the cell cycle is for one cell to become two cells, called daughter cells. To do this, the cell has to duplicate its DNA and split in two. The cell spends the first phase of the cell cycle, known as the G1 phase, growing. But at the end of this phase, it comes upon the G1 checkpoint, also known as the restriction point. Here, it has a big choice to make. If the cell decides to take a break from cell division, it will enter the G0 phase. An important protein known as p53 will activate another protein called p21. p21 prevents the cell from moving forward in the cell cycle by blocking the action of proteins known as cyclin-dependent kinases, CDKs for short, which are responsible for continuing the cell cycle. But if the cell decides to proceed with cell division, it must pass the inspection of the restriction point. The restriction point is where the cell makes sure that it is ready to divide by checking to make sure that the cell has enough proteins and organelles to go through with mitosis, and, most importantly for our purposes, it checks the DNA to make sure that it isn't damaged. Normally, a mutation in the cell's DNA is detected at the restriction point, and the cell repairs it. If this fails, most cells will move into a permanent G0 phase, known as senescence, or commit cell suicide, known as apoptosis, to prevent the mutation from being passed on. But supposing a mutation affected the function of the restriction point, what then? Well, then we have a problem. Without a gatekeeper to halt the cell cycle, the cell is left free to pass into the cell cycle before it is ready. The cell begins recklessly racing through the replication process over and over, and since it is no longer properly checking for DNA errors before it divides, those uncorrected errors begin piling up. Pretty soon, that one cell with one DNA error has turned into a whole mass of cells with innumerable errors, and, if this goes on, the cell will acquire all the right mutations to become a cancer. Any mistake that influences the sprawling complex of molecules that make up the restriction point can be the beginning of a cancer. Scientists have identified many genes where damage increases the risk of cancer, and divided them into three general groups. Proto-oncogenes are genes, the mutation of which can create a molecule that messes with the cell's ability to control division. Tumor suppressor genes are genes that normally halt cell division, but when mutated are unable to do their jobs. DNA repair genes are genes that do exactly what you would expect, repair DNA. A mutation to one of these makes it harder for the cell to fix its DNA, and so results in a higher rate of uncorrected mutations and thus a higher possibility for proto-oncogenes or tumor suppressor genes to be mutated.